confocal microscopy because we can use fluorescent tags and we can label those fluorescent tags on certain proteins that we're interested in studying. And it really labels up and makes the cells like glow different colors. And this is how we can see where things are localized, what they do. This goes even really, really high power. This is an example. This is an image I took. Any of you guys ever been in my office? Only a few of you have. So come by my office sometime and hang out, and especially for the exam, studying for the exam, or after the exam if you want to go over stuff. You'll see stuff hung all over my walls, and it's all artwork that's science, microscopy, images of things that I took. Because to me they look like art, not just science. This is an embryo. This is actually a mouse embryo hatching out of its little shell right here. But I stained it with some different markers so I could see all the nuclei, the nucleus of each individual cell in blue, and then I could see the plasma membranes in green. So I could sort of track it and what it was doing just under the confocal microscope. So all kinds of beautiful stuff we can capture. Oops. Here's some other examples. Light microscopy, this is staining so we can see the layers of cells and tissues. This is electron microscopy where we can see inside of those cells. This is scanning electron where basically you can see beautiful surface details. We can see cilia and sort of hair-like structures. This is lining the inside of your trachea, and this allows dirt and debris and particulate matter to be sort of swept up and eliminated, like when you have to hawk a loogie, or you get boogers, right? All the hairs, these cilia are helping to collect that stuff and prevent it from going down inside your lungs. So we can image all these things with microscopy. So the cells, this is a cartoon sort of rendition of a cell, or what a generic cell looks like. It's got all of its structures, the nucleus, the ER, we're going to look at, the Golgi. You know what these structures are. What are these little kidney beans? Mitochondria, good job. You know about what the mitochondria does already, right?
So the cell.
Yes, powerhouse of the cell. Okay, good. And you know the 3 metabolic pathways that feed into the mitochondria? What's the first one that happens in the cytosol? It actually happens out here.
Glycolysis.
First, where you break down sugar, the first step.
Glycolysis.
Glycolysis, and then Krebs cycle, and then
ETC.
you guys are on. You got it. You got that section, you're good. You just got to know what comes in and what goes out. That's the only other part. All right, so this is the overall cell. You know a bit about this already. We're going to spend some time on these other structures so you're aware of what the organelles do in the cell. Cells are really, really diverse. In our human body, there's about 200 different types of cells. That means they have unique anatomy and physiology. They have a morphology or a structure that makes them very specific to do a specific job. These are some cells taken, pictures of tissues and cells taken by some of my students. So in anatomy and in physiology, we do a lot of microscopy, and we look at human tissues and cells under the microscope. And whenever they get beautiful pictures, I ask if they can send them to me, and I keep them and use them. But this is just an array of different tissue types in the body made up of all these different cells. They actually took these with their phones, with their iPhones or their Androids, put them right up to the ocular of the microscope, held perfectly still, waited till that flash of light was right at the ocular of the phone, or the eyepiece of the phone, and then snap a picture. And I'm always, bless you, I'm always impressed because they just keep their phone handy, they keep it charged, and they can take tons of pictures. It helps them learn the structures. Their publication quality, I always tell them they're beautiful. This is cartilage, fibrocartilage. This is a tendon. This is bone. This is the gut, the GI tract, the large intestine, and that's the skin. So these are all just beautiful examples of all the different cells and tissues. What I want you to get from this is the cell isn't just this big blobby cell with all these structures. Cells get highly adapted and a unique morphology where some are long and skinny, some are flat and smashed, some are boxy and round, some have hairs on them like in the trachea. There's a beautiful diversity of different types of cells. We've got cells that allow you to move body parts. So they move parts and organs in your body, the muscles all in your body, skeletal, cardiac, and smooth. Those are the 3 major types of muscle that you have. And all these types of muscle cells or muscle fibers contain these proteins, these big long proteins called actin and myosin. These actin and myosin fibers of protein actually slide past each other. As long as you have ATP, if you have ATP to do energy, to do work, they slide past each other, so it causes muscle fibers to shorten. Okay? Muscle fibers contract. That's what we say. Excuse me. I'm burping. I'm sorry. The muscle fibers shorten. That's what happens when they're contracting. So if you're going to pull 2 bones, change the angle between them or move one with respect to the other. My muscle is actually shortening to pull that bone or that lever towards itself, right? So these particular proteins, actin and and myosin are these long polymers, and they actually, with energy, are able to slide past each other and allow your muscles to contract forcefully. And I would say this process of contracting, whether it's the heart pumping with every single beat, or whether it's the skeletal muscle fibers shortening to allow your body to move and your limbs to move, all of these processes require calcium and ATP. If you don't have enough calcium and you don't have enough ATP, you cannot do work with your muscles efficiently. Okay? So they're really important. We also have cells that cover and line the body in different organs. These are examples of covering cells that cover and line tissues, the skin, the trachea, and then the gut, like the small intestine. These cells are long, like columns. These are long, these have hairs on them, or cilia. And these cells are kind of flat and squashed. The surface ones are dead, actually. All the surface layers of your skin are completely dead. They're dead packed cells that have a waterproofing protein in them called keratin. It's actually in your hair, too. You know they do that keratin treatment, that Brazilian blowout thing, if you want to straighten it, right? They're actually adding keratin, so it straightens out. You have keratin packed into the surface layers of your skin. This is the superficial or outer layers of the skin, and this is as you go deeper. So all of these are types of cells that line and cover organs and your body itself. We also have cells that store nutrients. You should be familiar with these already, right? Fat cells, or adipose cells, store energy reserves, and they're good for a rainy day or when you don't have a lot of incoming nutrients. Fat adipose cells, adipocyte is the individual cell, like here's one, here's one, here's one. They store fat. The shape is made by that large fat droplet, so there's a contained little droplet that's in there, usually packed, filled with a lot of triglycerides, fat, in their cytoplasm. And their nucleus, actually, it's hard to see here, but I'll highlight one. So you can see, like, here's this cell. Its nucleus gets pushed over to the edge because the fat droplet, all the fat is filling up that whole area. So when you have a lot of excess stores, you've consumed a lot of fat or a lot of excess calories, that triggers the fat cells to create fat, store them, and they get filled with fat. So they increase in size. So when you gain weight, you're not gaining a whole bunch of new adipocytes. You're actually getting more fat within the adipocytes. So they can get scrawny and scraggly and not very full, or they get really full, packed with triglycerides, ready for fasting or when you need them. We also have cells that fight disease. So macrophages, I should have gotten a real one, this is a cartoon one. They move through tissues to reach infection sites, and they engulf foreign pathogens and microbes and particles and debris and dying cells. These guys will just go around by amoeboid action and just consume invaders and dying cells and parts of cells. Part of your immune system, actually. And macrophages are a type of white blood cell, and we're going to look at these in lab. So macrophages come from monocytes, which is a type of white blood cell. We're going to look at 5 white blood cells in lab tomorrow. We also have cells that carry gases. What are those? What are these?
Oh, red blood cells.
Red blood cells. Do you know the scientific name for red blood cells? We're going to study these in depth tomorrow in lab. Erythrocytes. Erythrocyte is a red blood cell, okay? It's E-R-Y, erythrocytes. And we have leukocytes too. So erythrocytes. Whenever you see the word cyte, that means cell. So this is a red blood cell. And then we have, leukocyte. These are white blood cells. So these are RBCs, very important. And these are white blood cells, very important. These basically carry gases, essential gases throughout your body. And these basically fight infection and disease. So part of your immune system really is your leukocytes or your white blood cells. We're going to do counts of these and analyze these and look at these under the scope all tomorrow in lab, which I find it's always pretty fascinating. These cells are really small. These are about 7 microns in diameter, so they're some of the smallest cells. Remember, they can barely get through capillaries. Some of the smallest vessels of your body, they can barely go through single file because they're teeny tiny. Okay? They're very important.
The red blood cells or the white?
The white blood cells are bigger. These are about 2 to 3 times the size of an erythrocyte or a red blood cell. And you'll be able to see under the microscope when you look at them. White blood cells are way bigger. Erythrocytes are tiny little biconcave little discs that are specialized for carrying gases. And the interesting thing about red blood cells too, they have no nucleus and they have no organelles, the structures that we're going to talk more about. They get rid of all of their organelles and their nucleus. They shed them, they exude them, get rid of them as they're developing in the red bone marrow. So that all they do is transport around tons of hemoglobin, which is carrying the oxygen and the CO2 back and forth from the lungs to the tissues. So they don't have mitochondria. They don't have a nucleus. They don't have a lot of these structures. So red blood cells are unique in that way. Yeah, Jack?
Okay, so like when you get sick, does your body produce more white blood cells, or
Yes.
is it just like?
Yes. Yes. So that's exactly right. So white blood cells start to proliferate. They get triggered to divide by mitosis and make more of them when you're exposed to specific infection, specific bacteria, specific pathogens, parasites, viruses. Certain types of white blood cells will proliferate. So you'll have more. And you can count those. We can do lab tests, and we know if there's an infection, if your white blood cell counts are elevated. Go up, there's some sort of infection. Got to figure out what it is. We're going to do that tomorrow. Yeah, it's very fun. Yeah.
But the same isn't true for fat cells, correct? Those don't multiply, they just fill up and
Exactly.
expand?
Exactly.
Okay.
So as Melissa said, the fat cells, they don't really multiply. They just fill up with fat or they release their fat. They fill up with fat or they release their fat, depending on the nutrient status and what your body needs, the demands, the caloric requirements of your body. So liposuction, that's one little thing related to the fat that Melissa just said. You don't gain more. They don't mitotically divide a whole bunch. They can, because there's stem cells in your fat pockets in your body. So if you suction them out, the stem cells in that area go, oh, we got a big hole, we got to replenish the hole, let's make some more adipocytes, and it kind of fills that in. It doesn't always fill it in the right way either, so it causes dimpling and weirdness. Have you ever seen like the lipo gone wrong and those different things? That's why. They suction out the cells, and the stem cells went, we need to make more, and they just filled them in sort of an irregular way. So when you lose fat, all you're doing is letting your adipocytes release their triglycerides, and you're burning them, you're metabolizing them. That's all. Instead of storing more, you're releasing more. We also have cells that transmit and gather information, like neurons, examples of this big long neuron that we have in the brain and in the spinal cord and in all the nerves of your body. This is all for receiving and transmitting messages. Here's some in the dish. I cultured these in vitro and labeled them with blue and red so I could see their processes and their nuclei and actually image them in vitro or in the dish. We also have cells for reproduction, and these are really interesting. We're going to spend a whole section on the menstrual cycle and fertilization and embryo development, becoming boy and girl, so we'll go into all this stuff. But we have eggs, we have fertilization. This is a crazy hyperactive sperm trying to get to the egg, and then fertilizing if it's successful. The one sperm fertilizes the one egg. You get the nuclei that fuse together. So this is the early one-cell embryo, or zygote. But that comes from that one sperm and that one oocyte or egg. So they're both really specialized. The sperm has a long tail or a flagellum, and it's made to swim, find, and fertilize the egg. So it's anatomy fits its physiology and what it needs to do. The egg, or the oocyte, is a large cell, the largest cell of the body based on diameter of the actual circumference of the cell, that has extra stores of RNA, extra stores of protein. So it's chock full of these nutrients and enzymes and RNA that are all right there and waiting. And it has a specialized outer shell that's made of glycoprotein, so it's this hard sort of shell. Has all the potential to give rise to all the cells and tissues of the body. If you think about it, it's pretty amazing. It's pretty amazing that one cell can give rise to every single other cell of the 40 trillion cell individual with all 200 sort of different types of specialized cells. It's really, really amazing. All that's needed is the trigger from the sperm and the delivering the chromosomes or the nucleus from the sperm to actually trigger embryo development. Okay, bless you. So cell structure, where we'll go through the different pieces or parts that make up cells, starting with the outside and then going to some of those organelles on the inside. We have the plasma membrane, okay? So this is a phospholipid bilayer. What is a phospholipid? At this point, you should sort of know what that is. It is a fact. It's a lipid. That's right, Simone. Yes. Phospholipid. We actually looked at it. It has a little choline group. It has a phosphate group, a P with a bunch of oxygens, and it likes water. And then it's got two fatty acid chains that hang down that don't like water, that are hydrophobic. Remember that? So it's amphipathic. It kind of, the head likes water, the tails don't like water. Remember that?
Oh.
Okay, be able to recognize what that is, looking at it, okay, and know its overall structure, because that's what makes up the cell membrane of every single one of your cells in your body, the plasma membrane, okay? It's a fluid mosaic in that it has a lipid bilayer. It's got 2 layers of those phospholipids. And do you guys remember how they line up? Remember they kind of go like this? That's what we say by bilayer. That's what we mean. It's got 2 layers. The heads, right, the polar heads with the fatty acid, the fat part that gets sandwiched in the middle. Remember this?
Yes.
Okay? This is what makes up the lipid bilayer. And this naturally will come together because like goes with like. These like the charged outside, those like the non-charged fatty interior. So this makes up the phospholipid bilayer of your cells. And it's a fluid mosaic model in that it's very fluid and it's got proteins inserted in it, it's got channels, it's got receptors, it's made of fat and protein interspersed. So it's not rigid, okay? It's not like a hunk of hard fat. It's fluid, it's lipid and it's fluid, so it can move, stuff moves around in it. The cell's very pliable and resilient, it can move. It doesn't stay hard and stationary like a plant cell wall, okay? So that's what gives it its chemical characteristics. And what type of lipid? You already answered. It's the phospholipids. So we looked at those phospholipids in the beginning when we talked about lipids. It's got a lot of proteins in it, integral proteins, peripheral proteins, receptors. All of those are embedded within the phospholipid bilayer. So if we look at it, there are little sugars coming off. These are basically little sugar molecules that help give the signature of the cell. So when cells get close to each other, they've got receptors, they've got proteins embedded in the lipid bilayer, and they've got this helix or kind of halo of sugar. That is like their fingerprint, so when they touch near another cell, they can kind of figure out the identity of other cells when they're close together. We've got integral proteins, and we've got peripheral proteins. Integral, if you integrate, like our integral program, you integrate and you're right in the middle of something, right? You go all the way and you're surrounded. You are integrated fully into that. Integral protein goes all the way through the plasma membrane, okay? Integral protein, integral protein, integral protein. Peripheral protein, if you're more at the edge, you stay at the periphery. These are proteins that are down here and they're kind of anchored to the skeleton filaments inside of the cell. So they're peripheral proteins, they're out on the outer part, they're out on the inner part. They're involved in anchoring or structure of the cell. Really, the integral proteins are the most important, and those are really the ones that stand out because those are different types of receptors, they're different types of channels. Those are what allow your cells to respond to the external environment when it comes across a chemical or another cell or a pathogen or something. The receptors and these integral proteins allow the cell to behave the way it needs to behave. Also taking in materials and letting out materials, like ions, for example, like calcium or sodium, okay? So these are really important. And these help to make the cell selectively permeable. So you've heard that language before, right? The plasma membrane is selectively permeable. That means it selects which things can get in and which things can get out. Not everything can just cross. You don't want that, right? Just like your skin on the outside of your body. You don't want everything penetrating through and getting through to the core of your body. So it's like a casing that's selectively permeable. So the plasma membrane acts as a barrier against substances outside the cell, so it can allow in selectively which ions and molecules need to get in. It allows cell-to-cell recognition and communication. It has receptors. So those integral proteins and different types of proteins can act as receptors. The cell can bind to different chemicals or bind to different cells and be able to recognize those things. And it can determine which substances enter and leave the cell, so it's a barrier. It's a selective barrier. And we say it's selectively permeable. Permeable means to permeate, to go through, right? It's selective. Not everything's getting in, not everything's getting out. I have a little video demo. I don't know if this will, she kind of goes through the chemicals that go through.
The plasma membrane is selectively permeable. It lets some substances through and not others. The hydrophobic interior of the bilayer is one reason membranes are selectively permeable. Hydrophobic molecules, those that are soluble in lipids, can
Fat.
easily pass through the membrane.
Fat can go
In addition, small molecules like O2 can
Gas.
sneak between the phospholipids of the membrane.
It's small.
On the other hand, large hydrophilic molecules like glucose
Sugars.
and ions, such as sodium ions
Charged.
and hydrogen ions,
Thanks.
cannot pass through the membrane unaided.
Did you see that? So those are kind of core principles about how the membrane is selectively permeable. If there are no channels or pores to get those things through, they're not getting through, okay? Because things like sugar, like glucose, are charged. That does not go in a fat, right? Fat and water or charged materials do not go together. Things that have a charge on them, like an ion, like sodium, it's not going to go through a fat. It has a charge. Those are polar. So things that are polar are not getting through, and things that are too big are not getting through. Now, if they're nonpolar, little fats, medium-sized fats, small fats, gases, things like oxygen and CO2, those are small and they're kind of lipid soluble, they go right through the plasma membrane, which is good. We want to get O2 in the cells and we want to get CO2 out of the cells because they're metabolically active all the time, right? But if we want to get sugar in the cell and we want to get ions in the cell, they need transporters or carriers, pores, so they can actually enter and leave. Yes?
For the things that get
What's-
rejected from permeating through, where do they go?
Just out in the internal of the extracellular space. So they are either in the capillaries in the blood or in the extracellular environment. And if they don't get taken in the cell, that's it. They're just not in the cell. They're carried on. And another cell might take them in. So a lot of times if the demands of the cell are it needs sugar, okay, it's starving, it's hungry, it has carriers that it'll put in the membrane and allow that sugar to be taken in through the carrier and transported into the cell. When it doesn't need the sugar, it doesn't put the carriers, it doesn't insert the carriers into the membrane. So it's selectively allowing in and out what it needs, okay? So, and cells are operating all over the place all the time, exactly what they need and what they need to take and give. Membrane transport, and that brings us exactly to what goes in and what comes out. Yes, Jackie?
Okay, so when the, like, say they need sugar, do they, the carriers that go grab them, do they break it down into smaller pieces, or is it like really little?
Nope.
Okay.
Glucose is small enough. It's a little ring structure. Remember, it's got little hydroxyl groups coming off. It's a little 6-carbon ring structure. There are glucose carriers, and we'll talk about these more when we talk about insulin and glucose very soon with the endocrine system. But basically, it gets transported into the carrier where it's open on one side, the glucose fits in there, and then it opens on the other side and lets the glucose go right in.
Okay.
But that's a good question. Once it gets in, that's when glucose starts to be burned or broken down through the metabolic pathways that we talked about, right? Okay, so some membrane transport. These are some concepts that come in handy when we talk about hormones and transport and ions and things throughout the body. Simple diffusion is the tendency of molecules to move down their concentration gradient. Down their concentration gradient means where they're concentrated or where they're most high in abundance, they go from that area to where there's less of those molecules. They're lowest in abundance or concentration, right? A good example is like cream. Have you guys drink coffee or tea or anything? Or when you put cream in there, what happens to the cream? Like if you were to just watch it, what would happen? Makes your coffee lighter, right? So it goes from where it's most dense with the cream and the sugar and the lactose molecules, the sugar, right? And it goes from where it's very dense and it kind of spreads out. It goes from high to low concentration. Now, I don't have an hour and a half to sit there and watch my cream diffuse. It's a nice experiment, but I grab a stick, right? I swirl it up and I'm on my way. Okay, so I just sped up diffusion. Kinetic energy and movement, and I increase the rate of diffusion. But it's basically just going from high to low. And it's natural because you guys probably all know this. You may not know why you already know this, but things will go from where they're dense to where they're least dense. There's more molecular motion and things are hitting each other when they're closer together. Hitting each other and bouncing off to where they're less dense in space, right? And so they're going to keep doing that until they even out and you have this equal distribution of molecules in that area, okay? So it's when you're close packed together, you're going to jostle around and bump into each other more until you actually spread out. That's all it is. Osmosis is specifically referring to diffusion of water, where the water molecules can go. And they actually do the same thing. This is diffusion of water molecules across a membrane, specifically the plasma membrane, as we'll talk about it, down water molecules gradient, okay? So this means water is going to go from where water is highest in concentration to to where water is lowest in concentration. Sounds weird, but it will make sense in a second. So if we look at, here's diffusion of a solute. So this is a solute like sugar in water. Okay, so we've got this membrane barrier here. This is supposed to imitate like the plasma membrane. It's got little pores. Pretend those are little sugar carriers, like allowing the sugar to move. If we have higher concentration of those sugar molecules on one side, and a lower concentration of that sugar in water on the other side, where is the sugar molecules? Where are they going to diffuse? Where are they going to move to?
Lower.
They're going to move to the lower side, hence the darker bold arrow going that way. Net diffusion is going to be mostly in this direction. Now notice there's still a skinny arrow going the other way. There's still random molecular motion and it going through pores, bless you, and going the other way. Just it's predominantly this way until it's going to be able to equal out. Okay, so ultimately the concentrations are going to level out on both sides, making that no net diffusion because they're going this way and they're going that way in equal proportions. Okay, so that's basic diffusion of a solute across a membrane in solutions. So if we talk about osmosis, water molecules don't carry a charge. And by the way, you have to know the chemistry of water very well. Okay? Now, I ask you lots of questions about water. What are the bonds that make up the internal water molecule? What are the bonds that connect one water molecule with a bunch of adjacent water molecules? What are the unique properties of water? So water is what makes up about 70% of our body, and it's a great solvent. It breaks things down, it carries things, it does all the things we need it to do. So water molecules don't have a charge, so they can pass through the membrane, but slowly, because they are polar, right? So they have the partial electronegativity of the oxygen, the electronegativity, and a little partial positive on the hydrogens. So they are polar, but they don't carry an actual charge like an ion. So they will kind of diffuse through the membrane slowly. But if we want to get them through a lot more quickly, your cells actually use these pores called aquaporin channels. And this aquaporin channel here actually lets water molecules go through at a more rapid rate. Okay? So water molecules, typically, if you want to move a lot of them in or out of the cell, which your cell does a lot of, many cells do, you're going to need an aquaporin or a channel to help mediate those polar molecules to go through this fatty lipid bilayer. So osmosis, the diffusion of the solvent, is what we're talking about, which is pretty unique. So water itself diffusing, so the solvent itself diffusing. And that's given a special name because it's a special process, and that's called osmosis. All right, so this is aided by channels in the cell's membranes, and they're called aquaporins. Aqua means water, porin is like a pore. Okay, so if you want to allow a lot of osmosis to happen, those particular cells have to insert a lot of aquaporins into their cell membranes. Kidneys, your kidneys have a bunch of these because they're filtering water all the time. All of your blood is being filtered by your kidneys. So in your kidneys, in your eyeballs, where there's lots of fluid in your actually eyeball itself, the lungs, salivary glands, and the brain, there's lots of aquaporins to allow this water to move. Now, water movement in osmosis, the net movement of water goes from the side with more water to the side with less water. So you might think, okay, this is super basic. I get this. It makes sense. Good. Hopefully if you do. But this is actually a little trickier than you think. So it goes from the side with more water, hence more dilute side, okay? So with less stuff in it, less solute in it means there's more proportional water molecules, and it travels to the side with less water, or the less dilute side, the side that has more solutes in it, more stuff. Does that make sense? Okay. So water's still going from high to low, it's still diffusing, but it's just this slightly different process that we're describing. Water moves from an area of low solute concentration. Whenever you see brackets in science, that means concentration. Low solute concentration to an area of high solute concentration. Water moves towards the side with more solutes, okay? So if you have water in a compartment or cell, and then water in a solution or a surrounding area, the water is always going to go from where it's more high in water molecule concentration to where it's less high in its concentration, or towards the stuff. The way I always remember this is water goes towards the stuff. If there's solutes or there's stuff in a solution or a compartment, the water is going to go into that area. Okay? Because wherever the stuff is, there's less water right there. Okay? Yes? Okay. So water flows downhill. down its own natural concentration gradient. So this actually shows this really nicely. It's going to show this little video. Little play. Yes? No, you didn't play. You have to hit the play button. Just make sure that this plays so nice. But it's not wanting to play. Okay, so I will tell you what it shows you. Diffusion, this is just net diffusion we just talked about. It evens out, the solid goes down its gradient. However, if you have a side that has a lot more solids, so I'll just draw it on here. If we have a lot of solids on one side, okay, but those solids can't move, okay? We've got no carriers. That membrane is not permeable to these molecules. Say there's sugar. We close it off. Boom, it's not permeable. No more carriers. Only thing that can move now is water, because we're assuming now the membrane is permeable to water. So in this situation, we've got more or less water over here. More or less water on the right side. More. More water because actually in this side there's more solids, there's more stuff. Proportionally, there's less water molecules because there's more solids in the same unit of volume. There's more stuff here. And actually water molecules will associate, they'll loosely associate and solvate those other little solids. So there's less free water molecules over here. Same unit, same space, more stuff, more sugar, there's less water. There's less water that's available over here. There's more water over here. There's less solids. There's like 12 solids here. Here there's like 50. Yes? Okay. Still with me? This is why people think osmosis, diffusion is very easy. It's not that easy. You have to really think about these concepts. So water is more highly concentrated here. It's free. It's not binding and solvating other stuff. It's free to do what it wants. Here, it is not. It's binding and associated with the solids, and there's more solids taking up more space. Where is water going to go in this situation? Okay, I'm getting hands all over the place. Okay, either this way or this way. Okay, this way? Yes? Okay, this way is the right answer. Good. Okay. Water is going to go from where it is high to where it is low. So this water line will go and move all the way up. Water will end up coming and coming and coming and coming until it's equal. Until I've got the same number of water molecules free over here as I have over here. Okay, this is why I want you to write down right now, water goes towards the stuff. Do you see why I said that? Water goes towards the solute. It goes towards the stuff. Because water is going from where it's high to where it's low. Okay? So when you think about water moving, you have to think about, okay, where are the solids? How many solids are over here? Is there a lot? Is there a little? How many over here? Because that determines where water is going to go down its gradient. This is very important with red blood cells, as we will talk about today and in lab tomorrow. How are we doing on time? Okay, we're good. All right, so tonicity. This comes into a physiology.